Hi students, today we are going to discuss about the skeletal muscle physiology. Actually, muscle mass or muscle tissue is made up of a large number of individual muscle cells or as a myosins. The muscle cells are commonly called as muscle fibers because these cells are long and slender appearance. Skeletal muscle fibers are multinucleated and arranged parallel to one another with some connective tissue in between that. So the muscular system function. First of all, you have to know about the functions. So it's going to be helpful to making the body movement and maintenance of the posture, respirations. In the respiration aspects, is the diaphragm and intercostal contraction, relaxation, and it's a communication for the verbal and the facial, and constriction of the organ vessels, like it's a peristalsis of intestinal tract, vasoconstriction of the and other structures and heartbeat and production of the body heat thermogenesis like that. So here, so it depends upon this muscles only they have to be giving the input to making the more function. So next of all we are coming for this properties of muscles is excitability, contractility, extensibility, elasticity. So excitability, which already seen this in nerve fibers also, I think so. So followed by that we are going to see also for the muscles is the capacity of muscle to respond to stimulus, and contractility, ability to muscles to shorten and generate the pulling the force, and extensibility is the muscle can stretch back to its original length. Elasticity is ability to muscle to recoil the original resting length after the stretched. And next we are going to see about the types of muscles. Like we have three types of muscles. One is the skeletal muscle, smooth muscle, and cardiac muscles. And but here we are going to see about only the skeletal muscles. First of all, we're going to see the skeletal muscle really attached to the bone and make up the 40% of the body weight, responsible for the locomotion, spatial expression, posture, respiratory movements, and other body movements. So voluntary actions controlled by the somatic motor neurons. So in the wall of the hollow organ, blood vessels, eye, gland, uterus, skin, some functions that propel urine, a mixed food in the digestive tract, dilating, constricting, and pupils are regulating the blood flow. So for the cardiac muscles, heart is a major source of the movement of blood, and as well as helps to making the autorhythmic and it's to control the involuntary by the endocrine and the autonomic nervous system. And next we are coming for to see the connective tissue sheets. So often have been made from the became like some muscles. So now we are going to see about there is a different as the steps, which means that we as a different layer which is covered like as a muscle. So connective tissue like as a muscle. So here, so what are the wrapping system which is we are going to see about that. First one is the epimysium. So actually, there's a beneath the fascia. Actually, this muscle. First of all, the muscle mass is be separated from the neighboring tissue by a thick fibrous tissue layer, is called the fascia. Okay, and then just beneath the fascia, muscle is covered by the connective tissue sheath is called as a epimysium. Okay, and next we are coming for to see this perimysium. So the connective tissue sheath that covers each fasciculus is called as a perimysium. Okay, and next we are coming for to see this endomysium. So each muscle fiber is covered by a connective tissue layer called as a endomysium. Okay, and next we are coming for to see this so collagen fibers of all three layers come together at each of the muscles form as a tendon or as aponeurosis. Next we are coming to see this how to situation on what are the things will be present in the skeletal muscle. So the long cylindrical cells, many nuclear cells, striated, voluntary, rapid contractions. These are things which occurs in the skeletal muscles. Next, we are going to see about the basic features of the skeletal muscles. The muscle attachment. Most skeletal muscles run from one bone to another bone. So one bone will move, other bone will be remain fixed. So actually this is origin and initiation is main. So origin is less movable attachment and initiation is a more movement attachment. So by this way this is going to be helpful to just making the movement. And following that is a muscle attachment to origin and insertion by the connective tissue, And the fleshy attachment as well as the indirect attachment. There's a two types of attachment which we present, and this connective tissue fibers are short and connective tissue. In indirect attachment, connective tissue forms a tendon or aponeurosis. Okay, and so mainly we are going to see about this muscle fiber are attached to the tough cord of connective tissue called a tendon. So tendon is term attached to the bone. Tendon of some muscle is thin, flat and stretched, but tough. Such type of tendon is called as aponeurosis. And each and every muscle fibers is enclosed by a cell membrane called plasma membrane that lies beneath the endomysium. It's also called as a sarcolemma. Okay, and now we are going to see about the structure. It's composed of muscle cell, connective tissue, blood vessels, nerves. The fibers are long, cylindrical, and the multinucleated. So it tends to be a smaller diameter in the small muscles and large in the large muscles. So 1 millimeter to 4 centimeter in length. And is developed from this myoblasts. Number of remain constant and striated appearance. Nucleus are peripherally located. Okay. And now we are going to see about this one in the attachments. So which already is shown in the previous slides. Next we are going to deal with this what is a muscle fibers. Okay, now we are going to see about this myofibrils first. As a myofibrils is or the parallel filament presence in the sarcoplasm of the muscle cells. Myofibrils are run through the entire length of the muscle fibers. In the cross section of this muscle fiber, the myofibrils appears like as a small distinct dots within the sarcoplasm. And in some muscle fibers, some of the myofibrils are arranged groups called as Bonheim areas or field. And it will be bundles of the protein filament like is called as a myofilament. And what are the two types of this myofilament we are going to see about this? One is actin filament, which is called as a thin filament, and another is a myosin filament, that is called as a thick filament. And attached end of the fibers muscle myofibrils are being anchored to the inner surface of the sarcolemma. When myofibrils is shortens, so muscles will be shortens, means the contract. Next, we are going to see about this Sarcotivases system. The Sarcotivases system is the system's membrane structures form the vesicles and tubules in the sarcoplasm of the muscle fibers. It surrounds the myofibrils, and the structure of the constituting the Sarcotivases systems it is two types. One is the T tubules and another L tubules. Other is called as the Sarcoplasmic Reticulum. Okay, first of all, we are going to see about the T tubules. Or the transverse tubules or narrow tubules is formed with the invagination of the sarcolemma, and this formed is the tubules penetrate all the way from one side of the muscle fiber to another side. That is, these tubules penetrate the muscle through through the and through the other muscles. Because of their origin from sarcolemma, the T tubules open the exterior of the muscle cells. Therefore, the VCF runs through the lumen. And next, we are coming for this sarcoplasmic reticulum. Is an elaborate and smooth endoplasmic reticulum. They run longitudinally and surrounding each myofibrils. And it will be just tubules or closed tubules that run in the long axis of the muscle fibers, forming the sarcoplasmic reticulum. These tubules form closed tubular systems around each myofibrils and do not open the exterior like T tubules. And as well as the L tubules, which means L tubules corresponds as the endoplasmic reticulum of other cells at regular intervals throughout the length of the myofibrils, and the L tubules is dilate to form a pair of the lateral sacs called as a terminal cistern. So each pair of the terminal cistern is in close contact with the T tubules. Okay. And in human skeletal muscle, the triads is situated in the junction between the A band and the I band that you're going to see in this slides. The calcium ions are stored, and when the circulation stores the calcium when the muscle is not contracting. And when stimulated calcium releases into sarcoplasms and then sarcoplasmic reticulum, the cell membranes as a calcium pumps the functions of pump calcium ions out of the sarcoplasm back into the sarcoplasmic reticulum after the contraction. Now this is a teeth tubules. Actually, in this teeth tubules, only the calcium ion have to be stored. So whenever there is making the contraction, it's going to just releases the calcium ion. They have to just fix it in this in the troponin. So they have once you have to fix it, they have to making the contraction. Then remaining parts again they come and stored in the teeth tubules itself. Okay, this is the tubules of the sarcoplasmic reticulum. And next we have to clearly have to explain how come it's going to be this is a microstructure of the muscle fibers. So this is a muscle mass, and here this is an endomysium, and this is a muscle fibers, and inside the muscle fibers we have to see about the myofibrils here, and inside the myofibrils we have as a sarcomere, and inside the sarcomere we have a actin and myosin band, and these two band only making the contraction. The entire muscle going to be make the contraction. And the sarcomeres is a disc too, is a disc. Sarcomere is a repeating the function unit of the myofibrils, about the 10,000 sarcomeres per myofibrils to end to end, and it is about 2 micrometer long. Actually, these two difference in size, density, distribution of thick and thin filaments gives the muscle fibers branded situation of the appearance. And next we are coming for to see this actin myosin. It is a most interior part of this microstructure of the myofibrils, and here there is a one is the actin, another is the myosin, and here these two only going to be helpful to just making the important function of to making the contractions. And first of all, we are going to see about the myosin. So what does it mean myosin? Which means a thick filament. So many elongated myosin molecules like the golf clubs. So a single filament contains roughly 300 myosin. And each myosin filament is consist about 200 myosin molecules. To about 18 classes of the myosins are identified. Actually, is each myosin molecule is made up of 6 polypeptide chains, which is 2 are heavy chains and 4 is the light chains. And here is going to be segregated as a tail portion as well as the head portion. The tail portion of the myosin molecule is made up of the 2 heavy chain, and head portion is one end as a double helix, and both the heavy chains are going to be turned away in the opposite direction in the form of the globular head portions. And in actin filament means a thin myofilaments be composed with 3 major proteins: fibrous actin, tropomyosin and troponin. So actin molecules are the major constituent of the thin actin filament. Each actin molecule is called as a F-actin, and is a polymeral as a small protein is called as a G-actin, and there are about the 300 to 400 actin molecules in each actin filament. The actin molecules in the actin filaments are also arranged in the form of the double helix, and each actin, which means the fibrous actin molecules, has an active site to which the myosin head is attached. And next we are coming to see this tropomyosin, and about 40 to 60 tropomyosin molecules are situated along the double helix strand of actin filament. Each tropomyosin molecule has a molecular weight around 70,000. In relaxed condition of the muscles, the tropomyosin molecules cover all the active sites of the F-actin molecules. And next we're coming to see the troponin. It is formed, and troponin is formed by the 3 subunits like the troponin I, troponin T, troponin C. Troponin binds to actin, troponin T is binds to tropomyosin, and it binds to calcium ions. Okay, and now putting it all together to perform the function of the muscle to making the contractions. So because it is initiated from the actin myosin filament, and the myofibrils have to be contracted, and after this muscle fibers and entire muscle fibers going to be contract. And next we are going to see about is the excitation contraction coupling. It's a muscle contraction, type of muscle contraction. It's a alpha motor neurons releases the acetylcholine. Acetylcholine produces a large amount of the EPSP and excitation potential and the muscles and nicotinic acetylcholine receptors and and the action potential excitations trigger the calcium ions releases to lead to fiber the contractions. The relaxations calcium ions levels to lower the organal reuptake.